Thank you very much, Cheryl. Welcome, everyone. I would like to congratulate you on coming to what I hope will be a very exciting day and a half. And the thanks for that goes to the planning committee that Cheryl mentioned. I really would like to thank everyone for the hard work that went into this. I can't tell you the number of conference calls that went into the planning, but I'm incredibly impressed with the amount of expertise that there is in this field, and we're going to have a lot of interesting discussions about the interface between human oncology and canine oncology. And I also wanted to thank IOM staff, particularly Cheryl and Patrick Ross, for their stewardship of us through this whole planning process and through the organization of this meeting. So I wanted to give a brief background to get us all on the same page about some of the challenges of cancer and how the interface between human oncology and canine oncology can benefit each other. And I'm sure everyone knows that cancer is a devastating diagnosis, but we frequently do not think about it as a public health problem. But it is a public health problem. 1 out of every 2 men and 1 out of every 3 women will be diagnosed with cancer in their lifetime. And there is no individual that is not touched by cancer that doesn't have a family member or a close friend that's been affected. Over 1.4 million Americans will be diagnosed with cancer this year, and over 600 million Americans will die from the disease. That all sounds pretty grim. On the other hand, we have over 14 million survivors of cancer. So we are making progress. We do have some success stories, but we have a long way to go. There clearly is a need for new therapies that are not only more effective at killing tumor cells, but are less effective at killing normal cells, so less toxicity. And so the process of developing new cancer therapies is unfortunately both slow and costly. It's been estimated that it takes an average of 13 to 16 years to bring a new therapeutic to the marketplace after the target has already been well validated. Much less than 10% of all drugs, once they've already been identified as a lead compound, ever make it to the marketplace. And we hear from our biotech and pharmaceutical company colleagues all the time about the cost of drug development, and it's been estimated that's on the order of $1.8 billion to bring a new drug to market. So we have a lot of challenges in not only the disease, but in developing new therapeutics for the disease. In addition, we'll hear today about many novel cancer drug candidates that fail in human clinical trials despite efficacy in traditional preclinical murine models. And we'll hear from Bev Teicher about the strengths and limitations of some of these murine models. But some of these that we frequently talk about is that they lack the key characteristics of human tumors in the sense that they don't have the long latency that most human tumors do. They don't have the natural causation. They don't often recapitulate the natural genetic instability of human tumors or the tumor heterogeneity, especially when we're talking about xenograft models. And then, of course, there's the tumor microenvironment that plays such an important role in human tumor development as well as responses to therapy that aren't recapitulated by a lot of the mouse models. So that's the story, and challenge of human cancer. What we frequently don't think about, at least those of us in the audience who are not in the veterinary field, is that cancer is also very common in pets. There's over 170 million pets in the US. Over 80 million of those are dogs. Almost half of all US households own at least one dog, and about 1 million dogs are treated for cancer in the US each year. So a statistic that surprised me was that cancer kills 50% of all dogs over the age of 10. So a very common cause of death. And it even kills 33% of dogs under the age of 10. And 33% of cats die from cancer as well. And we all know how motivated pet owners are. They're also motivated when their pets get diagnosed with cancer to enroll them in clinical trials. So that's an opportunity we'd like to take advantage of. And we'll hear a lot today about how the many characteristics that canine tumors share with human tumors. And this is particularly true in certain types of tumors, not all. Sarcomas, melanomas, gliomas, lymphomas are some of those examples. And some of the similarities include histologic appearance, some overlap in tumor genetics, and this is going to be a very important part of the discussion, especially as we move forward with targeted therapies in both human and canine tumors. So there's some genomics information that's already available about canine tumors, we'll be talking a lot about the potential gaps that we need to fill in order to make them more useful in designing new therapies. The biologic behavior of canine tumors, the molecular targets, therapeutic responses, acquired resistance, recurrence, and metastasis all share characteristics with human tumors, so a lot of opportunities to think about them in the same way. It's not just tumors that are shared between humans and canines. We all are familiar with the dogs who look like their owners, but there's a reason that we have a lot of shared characteristics. We've lived together for thousands of years. There really is this concept of convergent evolution in terms of the environment that we live in. And when one thinks about cancer development, that's not unimportant at all. Environmental exposures contribute to the majority of human cancers. And the other contributing factor is genetic susceptibility. It's always the interplay between the environment and genetic susceptibility that lead to development of cancer in humans. The same thing is true in dogs. And if we live in the same environment, there's a lot that can be learned about the biology of tumor development. We've learned a tremendous amount about cancer development in humans from, though they're rare, human cancer susceptibility syndromes. As we've identified the genes that are mutated in these familial syndromes, that's taught us a lot about the biology of how tumors develop, including what important exposures there are in the environment. We can similarly learn a lot from the susceptibilities of particular dog breeds to particular types of cancer. So I think this is an opportunity for us to think about scientifically in terms of understanding tumor development. We're going to hear a lot today about the NCI Comparative Oncology Trials Consortium, which has been in existence for a little over 10 years. We'll hear from Chan Khan and others about this extraordinary infrastructure that we'd like to think about how to take better advantage of it. The infrastructure and resources of this consortium are to integrate clinical trials for pet dogs with naturally occurring cancers into the development of new drugs, devices, and imaging techniques for human cancers, and it includes 18 veterinary academic centers. And the real challenge is how best to integrate these clinical trial data into the human cancer research enterprise and to integrate the clinical studies for pets into the cancer research continuum. And this is what we'll be addressing today. Now, there's always issues of regulatory constraints and considerations, and we need to be aware that the FDA has already weighed in on animal clinical trials in at least one setting, and that's with the FDA Animal Rule, which is about the approval of drugs and biological products when human efficacy studies are neither ethical nor feasible. And basically, it's for testing under the Animal Rule is a surrogate for human efficacy and clinical studies when it can't be done in humans. That does not do away with the need for doing the safety studies in humans. Those still have to be done, but the FDA can approve a product once human safety has been established and the animal rule requirements are met. In other words, it's based on adequate and well-controlled animal studies, the results of which establish that the product is reasonably likely to provide clinical benefit when administered in humans. There has to be a reasonably well understood pathophysiologic mechanism. The animal study outcome has to be clearly related to the desired benefit in human, which is virtually always reduced mortality or morbidity. The data on pharmacology parameters of the product in animals and humans allow selection of an effective dose in humans. It does not apply if product approval can be based on standards described elsewhere in FDA regulations, and it does not apply if you can do the efficacy trial in humans. And importantly, it's not a shortcut to approval, but it already shows us where the FDA stands on the use of information gained from animal clinical trials, and I think this will be an important consideration for our discussions over the next day and a half. So at this workshop, we're going to talk about the challenges and opportunities in cancer drug development. We'll talk about the state of canine tumor biology and genetics and where the gaps are and what things we can do to make them an even more useful model for understanding both canine and human tumor development and therapeutics. We'll talk about opportunities in imaging and other biologic endpoints that also impact on understanding human and treating human disease. We'll talk about the opportunities and challenges in the current human and canine clinical trials infrastructure, as I mentioned. We'll talk about the fact that we always have to consider that the pets are the patients in those animal clinical trials, just like the humans are patients in the human clinical trials. And so we have to ensure that the needs of the pet animals are addressed. And we need to think about how best to interface with the regulatory bodies and with pharma and biotech. So who will benefit from successful integration of canine models and human models? Human patients will benefit, canine patients will benefit, canine owners will benefit. It really is a win-win if we can do this effectively. It could lead to faster and less costly drug approvals if done correctly, and it could lead to better informed clinical trials design. And the way I've been thinking about it as we've gone through this process is we have two entities. We have the Canine Clinical Trials Consortium. We have many ways that we do human clinical trials for new cancer drug development, and they can inform each other. And we have many examples where that has been done successfully. They both can contribute to cancer drug approval. And those are drugs that are used either in the canine systems or in the human systems. Sometimes they will be the same drugs. Sometimes drugs that are developed in one system will end up being used in the other. We'll hear some examples of that, or vice versa. So I think we have to think about this as an iterative process. And the main goals of this, this day-and-a-half workshop is to think about how we can learn from each other and benefit everyone in the process. So thank you for your attention, and we'll start off with the first lecture from Dr. Lee Hellman.